So a very good morning to one and all. On behalf of Hindu College of Pharmacy, I am Dr. Konda Ravikumar, Professor, Department of Pharmaceutical Chemistry, and one of the conveners of current faculty training program to propose the inaugural address. A warm welcome to distinguished luminaries, delegates, faculty members, student fraternity from various colleges and universities for inauguration of 1 week international faculty training program and student training program on Dreap, drug research evaluation and practice. From 1st July to 6th July 2020, organized by Department of Pharmaceutical Chemistry in association with University College of Pharmaceutical Sciences, Acharya Nagarjuna University. In the present COVID-19 pandemic situation, the management of Hindu College of Pharmacy has given the extensive support to conduct the series of webinars from June onwards in various departments to enlighten the knowledge. I invite the August gathering our Honorable Chairman Sri Jupudi Rangaraju Garu, Honorable Secretary and Correspondent Dr. S. Masudan Rao Garu, Honorary Chairman Dr. M. Radhakrishnamurthy Garu, and Dr. A. Pramila Rani Garu, Principal University College of Pharmaceutical Sciences, ANU, our College Principal Dr. R. Govindarajan Garu, Vice Principal Dr. A. Sunita Garu, and OSD Sri P. Sitaramai Garu. Now, as a tradition, we will start with the lighting of lamp followed by a prayer song. Now I request our technical lead to proceed.
Shubham kurutvam kalyanam arogyam dhanasampadah. Shubham kurutvam kalyanam arogyam dhanasampadah. Shatru buddhi vinashaya deepajyotir namostute. Deepajyotir namostute. Shubham kurutvam kalyanam arogyam dhanasampadah. Shubham kurutvam kalyanam arogyam dhanasampadah.
Padmasana Ya Brahmachyuta Shankara Prabhrutibhi Devai Sada Vandita Sama Patu Saraswati Bhagavati Nishesha
My heartiest welcome to a great philanthropist, a renowned advocate of Guntur city, successful leader, able administrator, and moreover, Hindu College of Pharmacy Chairman Sri Jupudi Rangaraju Garu to this FTP. And I request Chairman Sir to address the gathering. Over to Chairman Sir.
Namaste. It's very informative. It is COVID-19, not only the education, but also the thrive of getting the faculty and the students coming up with good education aspect for the future. Our pharmaceutical department, headed by Govindarajan, has been doing good pickups for the students and the faculty. And I feel it that in the present scenario, the topic taken by our college is very much relevant, and our college associating with that of Acharya Nagarjuna University is a boom. Acharya Nagarjuna University is known for its credentials and applications and educational qualities. So the combination of Hindu College of Pharmacy and Acharya University, I think will throw a good light for the faculty development program and as well as students. At this hour, where there is no physical education, only distance education has been the process. But in the chemistry, there must be two combinations. So, the present system of education with that of the COVID-19 has become a chemistry for the faculty development program and as well as students. I feel that our college is trying to do whatever the best possible, especially under the marvelous guidance of our honorary chairman, Dr. Manara Krishnamurthy Garu, and our secretary correspondent, Dr. Ismail Sodarrao Garu, and all other faculty members who are doing their surveys for the benefit of the entire education and also for the development of good chemistical approach at this time of pandemic. I thank everyone and the participants who are participating. I feel that they will also have good knowledge, not only knowledge, but also try for the future chemistry for making out a good system of medicine, which the government of India and the world is struggling through this chemistical process. And I feel that this faculty development program and the student development program would throw a light on that aspects. I thank everyone, especially I thank the chemistry department, Govindarajan, Nagaraj, and others for doing that. Whatever little possible in sphere of education, our college is trying to do for the best. I thank everyone, and I feel that this one-week program will be a grand success. It will be annals of the history of our Hindu College of Pharmacy and ENU. Thank you very much. I feel that I must thank from the bottom of my heart all my faculty members who are doing their efforts in trying to bring up regions of faculty, regions of our pharmacy college, and especially Sunita has been doing a good job along with other members. I thank everyone. Bharat Mata ki Jai.
Thank you very much, sir, for your blessings and a grand welcome. I welcome a man of distinct vision and a fountainhead of illuminating ideas, an idol of knowledge and experience. He is none other than our secretary and correspondent, Dr. S. Masudan Rao Garu, a famous physician and surgeon. So thank you very much, sir, for your blessings. Now, my heartiest welcome to Dr. Manava Krishnamurthy Garu, retired professor of Guntur Medical College, founder secretary of Hindu College of Pharmacy, later chairman and presently honorary chairman and member of Pharmacy Council of India. So Dr. MRK sir, thank you very much, sir, for your wishes. I extend my cordial welcome to Dr. A. Pramila Rani Garu, principal, University College of Pharmaceutical Sciences, Acharya Nagarjuna University, and one of the convener of our Dreap faculty training program. I request madam to address the gathering. Over to you, madam.
Hello?
Madam.
Hello? Hello, can you hear me?
Yes, madam. Yes, madam.
Okay. Thank you, Ravi. Thank you,
madam.
Professor Ravi. Good morning to all participants, faculty, and students for this one-week international faculty training program on drug research evaluation and practice from 1st July to 6th July 2020. I congratulate the organizers, especially for the nice selection of the title for this one-week international training program. particularly during this COVID-19 outbreak, where the need for the suitable drugs synthesis or identification research to combat COVID-19, it is inevitable during these days, and one must learn about drug research and development and also various innovative technologies supporting R&D to come out with a specific result. And when you speak about historically, that means Indian medical history, man has explored nature to satisfy two major needs. One is food and another one is herbs for alleviating pain and suffering. And from then onwards, man has been searching for remedies to fight against diseases. And like in good olden terms or good olden days, herbs or mixtures of these herbs are being used to cure the diseases. And we all know that Ayurveda has been used in India for thousands of years. And in fact, our heritage, and we are rich in herbal therapy or herbal plants also. And later on, cumulative scientific and technological advances over the past 2 centuries have transformed drug discovery from old procedures to high-tech throughput processes of today. And I really congratulate the selection of resource persons for this 1 week training program, especially the innovative technologies that now the India has been following for the drug development and research, like bioinformatic approaches, and also the drugs by using bioinformatic approach, the synthesis of drugs for the treatment of COVID-19, and also computational drug discovery and screening technologies and molecular docking technologies, and other than these chemical approaches, ethical uses of animal models in drug research, drug design and discovery approaches, whatnot, everything that has been included in during these 6 weeks training program. And really, I feel very fortunate, and also it's my privilege to take part in this prestigious webinar, and that gives a very good benefit and enrichment in the knowledge of not only faculty but also students who have registered for this wonderful program. And I once again congratulate the management of the prestigious institution, and I always feel that this is one of the premier institutions in India, and also one of the very good affiliated colleges under Acharya Nagarjuna University jurisdiction. And also I once again congratulate the management to support this wonderful program, and also team Hindu College of Pharmacy principal and staff in general, and also the Department of Pharmaceutical Chemistry in particular. My heartiest congratulations to all the faculty and team Hindu College of Pharmacy who are involved and who are being taken pain to come or to conduct this excellent program during these 6 days. And I wish you all success. And once again, thank you very much for giving me this wonderful opportunity, and also making University College of Pharmaceutical Sciences as part of it. Thank you. Thank you, Principal Professor Govindarajan, and also Vice Principal Professor Sunita Madam, and OSD Professor Sitaramaya Garu, and other faculty in Department of Chemistry, Professor Ravi Kumar and others, and other faculty members from other colleges, other universities, and also students. And I wish you this program a very grand success, like the previous so many webinars that you have organized from May onwards. Thank you very much once again.
Thank you very much for your encouragement, best wishes, and continuous support always in all aspects, madam. Thank you very much.
Thank you.
I extend my cordial welcome to driving force of our college, moreover our principal, convener of FTP, Dr. R. Govindarajan Garu. I request Principal Sir to address the gathering and explain about the Dreap program. Over to Principal Sir.
Guru Brahma, Guru Vishnu, Guru Deva Mageshwaraha, Guru Saksha Parabrahma, Takshmay Shri Guruve Namaha. In this happiest occasion, I am extend my warm welcome all the luminaries of pharma fraternity, professors, scientists, industrialists, young faculty members, scholars, and wonderful students from various colleges and university in all over the countries for the inauguration of 1 week international faculty training and student training program on drug research, evaluations, and practice from 1st July to 6th July in association with University College of Pharmaceutical Sciences, Achar Nagar University. This is my privilege. I take immense pleasure in cordially welcome our, our respected chairman, sir, Juvid Rangaraj. He is able administrator, shows activities, leading the activities in the society, and also Hcop by pharmaceutical PhD spokesperson and present PhD meeting convener. He is exceeding the knowledge and support for development of our college. I kindly welcome sir to the great engineering our secretary correspondent Dr. S. Masood, Chief Physician and Surgeon of the Guntur City. My heartiest welcome to Mannava Radhakrishnan Garu, retired professor of Guntur Medical College, leading physician and diabetologist of Guntur City, founder secretary, later chairman and presently honorary chairman of Hindu College of Pharmacy and Central Council member, Pharmacy Council of India, New Delhi. Welcome to our respected governing body members, vice principals, pharmacy director, HODs, my dear fellow faculties, supporting staff, and students of Hindu College of Pharmacy in these inaugural functions. Now I extend my warm welcome to our senior professor of Andhra Coastal Region, Dr. A. Pramanarani Madam, Principal, University College of Pharmaceutical Sciences, Achar Nagar University, Nagar. And also welcome Dr. Ravishankar Reddy Garu, Assistant Professor, Achar Nagar University. In this junction, I would like to thank Honorable Vice Chancellor Professor P. Rajshekhar Garu and Honorable Registrar Professor K. Rosia Garu, Achar Nagar University, to support for this webinar. Hindu College is one of the oldest, renowned and recognized college in state of Andhra Pradesh. Hindu College of Pharmacy Hindu College of Pharmacy is governed by reputed Hindu College and High School Council, which is serving in the field of education since 160 years and managed by eminent professionals and philanthropists. Hindu College of Pharmacy was established in the year of 1999. It is affiliated to Achar Nagar University and approved by SBTT, recognized by ACT, PCI, and ISO recognized institutions. Our mission of the college to impart world-class education through state-of-the-art infrastructure and through traditional as well as contemporary ICT-enabled pedagogical practice. Our college and management to see the students goes out of the college not only with their degree, but with confidence, competence, caliber, and conviction and preparedness for any task that challenge is their own knowledge or career. The main objective of 1 week international EFTP and ESTP on drug research, evaluation, and practice. It indicates it's a Dreap. Dreap, it means drug reaping technology, because drug harvesting by using modern synthetic tools. At present COVID-19 pandemic outbreak situations, we have very much difficulty to identify the molecule to target the unknown disease. The strategy of the treatment on particular disease is now challengeable. In these situations, the selection of drug for the treatment of disease, either new entities or repurposing drugs only. So the purpose of drug discovery and development to establishment of activity is very much critical in the emergency conditions. In this platform, the topics which is chosen is enlighten the techniques and tools for the evaluation of drug synthesis and finding the methods for activity for unknown or upcoming disease. In this regard, we have invited we are organizing 7 plenary sessions for this 1 week faculty and student training program. All the sessions topics are linked directly or indirectly on the themes. I extend my gratitude to invite the speakers for this webinar for different institutes and universities from across India, Malaysia, and USA to discuss on theme of drug reaping methods, Dreap. Now, on day one, we are invited Dr. Anshuman Dixit, Senior Scientist E, Institute of Life Sciences, Bhubaneswar. And day two, Dr. Sudhir Verma, Principal Hydro Analytics and Bioinformatics Consultant from USA. And third day, Dr. M. Mallika, founder and instructor on Mass Training Academy and is former faculty of NIPER, Hyderabad. And fifth day, we are inviting Dr. E. Manivannan, Assistant Professor, School of Pharmacy, Devi Ahilya Vidyalaya, Indore, on third day, on fourth day. And also the same day, we have invited other speakers, Dr. Karthikeyan Chandrabose, Assistant Professor, Department of Pharmacy, Indira Gandhi National Tribal University, Amarkantak. And fifth day, we are invited Dr. Prasan Priya Nayak, Associate Professor, Department of Physiology, All India Institute of Medical Science, Jodhpur, Rajasthan. And sixth day, we are invited Dr. Vijay Kotra, Associate Professor, Faculty of Pharmacy, Quest International University, Perak, Malaysia. So now, I extend my hearty welcome to the speaker, Dr. Anshuman Dixit, Senior Scientist, E, Institute of Life Sciences, Bhubaneswar. He is a good teacher and scholar, my friend and ex-colleague at Jivaji University, Gwalior. He is also a good innovator. Is based on his innovations. He shared a link on recent discovery on identification of drug targeting multiple viral and human protein using computational analysis for repurposing against COVID-19. So already this linkage, his publications, we shared with all the participants in Telegram link. Please go through his achievement. I think so everybody is aware of that. Was the repurposing drugs against COVID-19. Now I request Mrs. Jansi to introduce our speaker. Wait. Now I request to over to Dr. Ravi Kumar. Next.
Thank you, sir. Thank you, sir. I request our intellectual, charming and active personality and moreover our Vice Principal, Madam, Dr. A. Sunita Garu, to address the gathering. Over to Madam.
Good morning, ladies and gentlemen. I feel privileged to extend my genial welcome to distinguished delegates, eminent speakers, and all the participants who came up with more enthusiasm. We, the management of Hindu College of Pharmacy, made strenuous efforts to maintain your enthusiasm by conducting series of webinars and faculty development programs for enrichment of knowledge in pharmaceutical realm. Every faculty might be having numerous degrees and vast experience in their field, though it's need to explore something unknown which helps to guide the students to face the upcoming challenging world. As we all know that the drug research is the process through which potential new medicines are identified. It involves a wide range of scientific disciplines, including biology, chemistry, ecology, and all other fields. Today, by taking a part in this session, let's grasp the innovative upgrade techniques related to research. I appreciate conveners of this program, Dr. R. Govindarajan, and Principal, University College of Pharmaceutical Sciences, Dr. Pramila Rani Garu, co-conveners, organizing secretaries for conducting this online faculty training program or student training program by inviting eminent personalities like Dr. Anshuman Dixit, Senior Scientist E, Dr. Sudhir Verma, and other delegates with a deep sense of gratitude. I express my sincere thanks to all the speakers who have accepted our invitation. I extend my sincere thanks to the management Hindu College of Pharmacy for providing such wonderful opportunities to all of us. I wish you good luck. Thank you for giving me this opportunity.
Thank Thank you very much, madam, for your nice words, encouragement, and support in all aspects. Now I request OSD and FAMD Director Sri P. Sitaramay garu to address the gathering. Over to OSD, sir.
Hello, good morning to all the viewers and my dear students, research scholars, and guests. I'm happy to address the gathering today on the occasion of the inauguration of the DRAP, which is initiated and conducted by our Department of Pharmaceutical Chemistry. And I congratulate Dr. Govindarajan and his team for selecting good topics and good speakers, not only from our Indian universities, but also from other countries like Bristol, USA, and Malaysia. And in this connection, I wish to emphasize that in the discovery of new drugs, the chemist plays an important role because they are the ones, the first scientists who are identifying the drug and giving the molecule to the further development. And our management is encouraging to conduct such seminars and webinars. And I must thank all our management members, our chairman, Sri Jupudi Rangaraju Garu, secretary correspondent, Dr. Sudosan Garu, and the honorary chairman, Dr. Manavar Radhakrishnamurthy Garu. In this connection, I would like to congratulate our Acharya Nagarjuna University principal, Dr. Tamila Rani, and other faculty members, Dr. Ravi Shankar, Reddy Garu, who are coordinating this seminar. And I wish to tell you that in Andhra Pradesh, the pharmacy college in Andhra University was started by Professor Rangaswamy, who himself was a chemist. And under his leadership, the Andhra University has started 60 years back the pharmacy course. And afterwards, he has gone to Delhi, and there he has started pharmacy in Delhi University also. And the Andhra University is managed by Professor Subbarao, Vishwanathan, Vishweswaram, and others. And since it is a pharmaceutical chemistry, I would like to remind one of the pharmaceutical chemists, Professor from Punjab University, Harikishan Singh, who has not only taught and guided the students, but also written history of pharmacy in various subjects. So I feel it is very happy to address you. My dear students, I request all of you to go through the textbooks written by Professor Harikishan Singh and services rendered by him. And in this connection, I'd like to tell you, in our country, the CSIR, Central Scientific and Industrial Research, have started 40 laboratories all over the country and states. And out of them, the best one I suggest for those who have not gone through is Central Drug Research Institute, Lucknow. They have got a lot of senior faculty who are continuing research in different fields. And especially for the students of B.Pharm and M.Pharm, I request them to go through the website of CDRI and know the development and take guidance from them. So wishing good luck for all of you. And today being the first Ekadasi, I wish Lord Vishnu will shower his blessings on all of you, and wish you good luck in your careers. Thank you, sir. Thank you very much.
Thank you, sir, for your best wishes and blessings. A fraternal welcome to all Hcop Hindu College of Pharmacy HODs, my fellow faculty members and students who play a vital role in the development of our institution. I request faculty Mrs. V. Jansi to introduce today's speaker, Dr. Anshuman Dikshit Garu, Senior Scientist E from Institute of Life Sciences, Bhubaneswar. So thank you all. Wish you good luck.
A very good morning to one and all. I am Jansi Rani V, Assistant Professor, Department of Pharmaceutical Chemistry, Hindu College of Pharmacy. We are very fortunate enough to have such an eminent personality as our today's speaker, Dr. Anshuman Dikshit. It's my privilege to introduce our today's speaker, Dr. Anshuman Dikshit, Senior Scientist, Institute of Life Sciences, Bhubaneswar. It is an autonomous institute of Department of Biotechnology, Government of India. Today, Sir is going to deliver the topic identification of drugs for the treatment of COVID-19 using bioinformatics approaches. Now, on behalf of Hindu College of Pharmacy, I am presenting a brief profile of Dr. Anshuman Dikshit. Sir has successfully, coming to his academic credentials, Sir has successfully completed his PhD from Central Drug Research Institute, Lucknow, India in 2007. Sir has completed his B Pharm, M Pharm and in from Department of Pharmaceutical Sciences, Dr. Harsingh Gaur University, Sagar, Madhya Pradesh. And Sir has published more than 50 research articles in various reputed national and international journals with a Google Scholar citations of 1,444 citations, having H index 24 and I index 35. Coming to his research potential, currently SAR has been working with the Institute of Life Sciences, Bhubaneswar as Senior Scientist E. SAR has 5 years of doctoral research experience at Central Drug Research Institute, Lucknow. And SAR is also having 4 years of postdoctoral research experience at University of Kansas, Lawrence, KS, USA. His research mainly on drug research and discovery and bioinformatics. His research areas includes identification of candidate genes related to oral squamous cell carcinoma and modeling and structural studies of Candida albicans DNA polymerases. Structure function analysis in copper zinc superoxide dismutase of Ipomia carnea plant. It is a flowering plant. Next, the modeling of docking studies on A3R receptors, adenosine receptors. This is one of the G protein coupled receptor, adenosine receptors, A3R receptors. Next research area of SARS includes virtual screening of chemical libraries to identify FTSZ inhibitors. FTSZ, it is a protein. FTSZ means filamenting temperature sensitive mutant Z. Virtual screening of these chemical libraries to identify these protein inhibitors. Next, last area that includes molecular modeling studies on TLRs. TLRs means toll-like receptors. These receptors act as a sensors for innate immune system. Innate immune system. Sir, thank you. With this, I conclude. Thank you, sir. Thank you very much for accepting our invitation. Thank you very much for giving me this opportunity. Now over to program convener, Dr. Govindarajan, sir.
So, whatever your shown this biodata is small update, but is a lot of activities, a lot of credits on his head. But we not show everything, but we really happy to say my friend Dr. Anshuman Dikshit with us on the platform this for first day. So is going to deliver on the speech of identification of drugs for the treatment of COVID-19 using bioinformatics approaches. Now I request to Anshuman Dikshit to start to deliver the speech. Now, the Anshuman, please share your PPT and make it your annotation to this.
So shall I go ahead? Post disabled participant screen sharing. So
Hello?
please enable,
Okay?
please enable my screen
Not,
sharing.
yes, sir. Now, my voice out.
Okay, so can you now see my screen?
Done. Kindly annotation disable. Yeah, under more option.
Yeah.
Okay. Kindly make annotation disable. Anshuman?
Hello. Hello. Can you hear me?
Hello? Yes, yes, sir. Okay. Yes, sir. It's okay.
Okay. So should I start?
So kindly disable your annotation. Under see your sharing and more option. Annotation is enabled. Now make it disable.
Okay, sure.
Under more option.
Done. That is done.
Okay, sir. Okay, sir.
Should I start?
Thank you.
Okay, so as I was introduced very nicely by ma'am, so she has already talked about my background, and my background is mostly computational biology and bioinformatics, and we are currently working on repurposing of drugs for COVID-19. And today's talk is our recent studies, or recent computational analysis on how we can identify drugs for COVID-19 using bioinformatics techniques. So in this current study, we have used multiple viral and human proteins. Viral means the viral proteins of novel coronavirus or SARS-CoV-2, and human proteins which participate in the infection process. So for any successful infection, there are two parts. One is the virus part, the other one is the host part. So we have tried to target both of the parts in order to identify molecules which can affect both of the processes. So before going on to the actual study, it's customary to introduce you about the problem at hand, although most of you already know about coronavirus and COVID-19 disease. In the last few decades, we have seen many viral epidemics. Notable among them are Ebola, Zika, Nipah virus. Nipah virus in 2013-14, we saw an outbreak in India. Then we have influenza H1N1. Then we have SARS, which had an outbreak in 2003, MERS, and current SARS-CoV-2. So what happened at the end of 2019? Mysterious pneumonia cases began to emerge in China's Wuhan city. Later on, upon investigation, when the people saw a large number of cases, investigation revealed that there is a novel coronavirus, which was named as SARS-CoV-2. And the disease was termed as coronavirus disease 19 or COVID-19. Now everybody is talking about COVID-19 around the globe. The major effects of this particular disease are SARS-like symptoms, that is the respiratory distress, fibrosis in the lung, and that leads to lung injury. So these two, the last two are actually the problematic one, the fibrosis in lung, and which leads to lung injury and ultimately respiratory failure. This COVID-19 is one of the world's worst pandemic, and especially in the last 100 years, it has been the worst pandemic. As of today, 1st July 2020, it has affected more than 10 million people globally. The average lethality, they say, is about 3%. In India today, we saw about almost 0.56 million cases as of today. That is 5,60,000 cases, which means we have to take this particular disease very, very seriously. And that's why a lot of institutes around the world, they are working to develop drugs for this particular disease. There are multiple efforts which are going on to develop vaccines and other strategies to tackle the situation. So my today's talk will be mostly on one of our study which we have recently published on ChemArchive, the identification of drugs targeting multiple viral and human proteins using computational analysis for repurposing against COVID-19. So repurposing means the drugs already exist for some other element, but they are not known to be active against COVID-19. So we have tried computational tools to see whether these particular drugs which are already in circulation can be active against this particular virus or not. So why we have used computational analysis? So the computational analysis have the power because the computers have enormous power, and these techniques can quickly identify molecules that can target viral proteins, and that can suggest candidates for repurposing. So we have about 3,000 drugs which are currently approved by US FDA, the Food and Drug Administration. And out of those 3,000 drugs, about 3,000 drugs, we tried to identify how many of them can bind to multiple viral proteins of this particular coronavirus. So what is the current status? The current status is there are global efforts, as I said, are going on, and many in silico studies have been done. Some drugs have been already identified which can bind to mostly the RNA-dependent RNA polymerase, RDRP protein, the spike protein, the membrane protein, the main protease, the 3CL-Pro, and other human proteins such as ACE2. ACE2 is the protein on which this particular virus latches first to get entry into human cell. This is the receptor for SARS-CoV-2. And among notable drugs which have been identified, most of them are actually antiviral drugs: zanamivir, indinavir, sakinavir, lopinavir, and remdesivir. Remdesivir is the star drug nowadays. Everybody is talking about remdesivir. It is an inhibitor of RNA-dependent RNA polymerase or RDRP protein. We'll discuss about these things later. So first we will start with the kind of studies we started. So we took the FDA approved drugs. Why? Because they can be quickly repurposed for the treatment of COVID-19. And our strategy, as I already discussed, we have used structure-based virtual screening, mostly docking, for the screening of viral proteins. The structures of the 3D structures of the proteins were taken from mostly X-ray crystallography and computational models. In addition, we also analyzed if these molecules can affect the host proteins, because as I said, the host and virus interaction is an important, means it is required for a successful infection. So we wanted to target both sides of the infection. So this is a general overview of how the coronavirus infection happens. And I'll talk about it very briefly. So on the top, you can see here, we have got a spike protein or S protein on the top of the screen, and it comes and latches on ACE2 receptor, right? The spike protein gives this particular virus a very unique shape, a corona-like shape, and that's why this virus is named as coronavirus. You can see in the picture. So it first latches onto the ACE2 protein. So the first target is ACE2 protein for the identification of inhibitors. If you can inhibit ACE2, the virus cannot bind to the cell surface. Number 2 is if you can inhibit the S protein or the spike protein, again the virus will not be able to bind onto the surface. Number 3 is the target when the S protein and ACE2 protein interface where they bind together. So if you can put some molecule which will prevent the binding, that is also a very nice target. So based on certain things, the knowledge of experimental things and biology, a lot of targets have been designed. So there are about 20 proteins which have been shown to be effective, which can be used as targets. There are 4 structural and 16 non-structural proteins in SARS coronavirus, and people have targeted many of these proteins in the endeavor to identify new drugs. So this is how it happens. So the virus binds to the surface, then membrane fusion happens, and once inside the cell, there is a replication complex which actually creates multiple copies of the viral genomic material, and this is led by the RDRP, the RNA-dependent RNA polymerase protein. Because a lot of students are there, so I will tell in a very common language. So this RDRP work like a photocopier. So it creates multiple copies of the viral genome very quickly inside the cell. And then what happens, there is a long peptide. The long peptide is generated, and this is cut into multiple functional proteins by the main protease. And there are some other proteins, which is PL, PRO, and others. So in this way, the virus replicates. And once the whole virion is there, then it will go out of the cell. So this is a brief mechanism of how virus enters and replicates inside the cell. Now, what we have done here is, you can see here there are 2 panels, panel A and panel B. In panel A, you can see there are proteins which were collected from protein data bank. These are the X-ray structures. Many of the proteins were modeled using computational homology modeling techniques, threading, and iTaser. And on these proteins, we have docked FDA-approved drugs. All the FDA-approved drugs, we have downloaded the structures, we have downloaded from the internet, and we docked all those molecules into different viral proteins. And what we have done here is, we took top scoring molecules for target proteins. So we have taken all those molecules which are showing at least a docking score of minus 8.5 using glide module of Schrodinger molecular model software. So here we have got, if you see below, we have got a heat map kind of thing. On the right hand side, panel B, we have got a data set from Gene Expression Omnibus, or GA, NCBI-GEO, which is a repository of multiple kind of expression studies. So we have taken gene expression data for 2 normal and 1 COVID-19 patient. This is the lung cell data. So we have got 2 normal lung samples plus 1 COVID-19 patient's lung sample. And then we try to identify those genes which are getting differentially expressed as a result of COVID infection. So from there we knew that how many proteins in humans they got differentially expressed as a result of this particular infection. And if we can target those proteins, especially those proteins which are getting up, probably some of them can be a good target. So we have identified certain proteins, and then we did a biological network analysis, which you can see on the lower right hand side of the screen. And then we tried to match both of the panels on the left and right side. And then we have identified some molecules which can target both viral and human proteins. So these are the 13 proteins which we have, the viral proteins or the proteins of novel coronavirus we have targeted. These are surface glycoprotein, membrane glycoprotein, nucleocapsid, NHP2, NHP3, NHP4, and up to NHP16. Of course, it includes the most important proteins, which is main protease, RDRP, and helicases, which are target of multiple drug discovery studies, spike protein and all. So these 13 proteins were taken, and you can see here the source, whether they were homologous modeled or whether they were obtained from protein data bank. Now, the first one is, as I said, when a virus comes to the host cell, so the first part is the attachment onto the host cell, and that happens by the interaction of viral spike protein and ACE2 protein, the angiotensin converting enzyme 2 protein. Almost all the pharmacy students are there, so I don't need to explain about what is ACE2 protein. Everybody knows about it. What are the ACE inhibitors? Also, people know about them. So, recent studies have provided experimental evidence that the spike protein of coronavirus binds to the ACE protein. So, as I said, these two proteins have become natural targets for antiviral drug discovery, and also the interface where these two molecules bind. So, if we can block that particular interface, that also is very nice drug target. So we have taken spike protein as our drug target because that is the viral protein, and we are talking about viral protein as drug target, which we have targeted here. And I'll show you how we did it. So we took this particular molecule to Schrodinger molecular modeling software. We prepared this particular protein using standard techniques, and then we docked all those drug-like molecules, FDA-approved drug molecules into this particular protein. And we retained all those molecules which were showing a score of at least minus 8.5. So here you can see the docking of one particular molecule, which is known as nebivolol. So this particular molecule is binding into the spike protein of coronavirus. On the right hand side, you can see the 3D image of the cavity where these molecules are binding. So on the left hand side, we have shown just one molecule. On the right hand side, we have shown multiple molecules which are binding inside the cavity. So here, if you see the 2D plot of nebivolol binding, so the molecule is shown in black sticks. The hydrogen bonds are shown in magenta color. So the bond coming towards the molecule, so they are showing the molecule is acting as a hydrogen bond acceptor, while those arrows which are going towards the receptor shows the molecule is acting as a hydrogen bond donor. So both hydrogen bond acceptor and hydrogen bond donor interactions are there, and there are of course some hydrophobic interactions with the phenylalanine of the receptor, especially if you see here 814, 824 are the hydrophobic residue where this hydrophobic part of the molecule goes into. And this molecule shows a very nice docking score of minus 8.5. So this is one of our hit molecules. Next is the M protein or the membrane protein of this is another structural protein, and it interacts with major major other structural proteins and viral RNA. It is 22 to 25 kDa transmembrane glycoprotein, and basically it facilitates the virion formation, the formation of virus particles and shaping of the viral envelope. So that's why it is a very important one. If you can block this, then the virion formation will be faulty and it can stop the viral replication. It also stabilizes the nucleocapsid. So if when it stabilizes the nucleocapsid, so therefore it can result in the proper shaping of the envelope. So this is another very important target, and we are showing the docking of one particular molecule which is known as cangrelor in N protein or nucleocapsid, the M protein of this particular coronavirus. And you can see here the cangrelor is making multiple hydrogen bonds on the left hand side. The molecule is shown in green sticks, while the receptor is shown in blue sticks. The docking score is minus 8.53, which is a very good docking score. That shows the molecule is binding with very good affinity to the receptor. And on the right hand side, we have shown 3D view of this particular molecule inside the receptor cavity. We have identified some other molecules as well, but this molecule, cangrelor, as we will show you, that this particular molecule binds to multiple receptors. That's why we have shown it here. The next one is the nucleocapsid or N protein. So this particular protein is very important for viral RNA packaging. That's why it is called nucleocapsid protein. It is made up of two distinct RNA binding domains, the N and C terminal domains. And these two domains are linked by SR-rich domain, or which is known as SRD domain. Previous studies suggest that this particular protein, N protein, inhibits TGF-beta, AP1, and Hcop signaling, and type 1 interferon production. Right? The sera of COVID-19 patient, and this is something which is very important, that the presence of IgA, IgM, and IgG against N proteins, that suggests it can elicit some kind of humoral immune response. So that's why this is very, very important protein. And in our study, we have found that ribavirin is a broad spectrum antiviral drug, vasopressin, octreotide, capreomycin. Capreomycin is an important drug. This is our anti-tubercular drug, which is used for drug-resistant tuberculosis cases. So very nice antibiotic. So these molecules bind to the N protein. And here we show the binding of nucleocapsid protein with capreomycin. So you can see again we have multiple hydrogen bond interactions of capreomycin with nucleocapsid protein, and the overall docking score is minus 8.8, which again shows some kind of strong docking score. And on the right hand panel, you can see the cavity view, where we show the receptor cavity, and there are multiple molecules which are sitting inside the receptor cavity. The next one is, and this is methyltransferase or NSP16. This is another important target for SARS-CoV-2, and this particular NSP16, this is an important protein, and this what it does is it protects viral transcripts from host exoribonucleases. Therefore, it helps in escaping the host innate immune response. Right? That is very important. That this particular protein helps in escaping the host innate immune response. So, if we can block this particular protein, probably our immune system can more effectively neutralize this particular virus. Right? Therefore, this particular NSP16 is a potential target for antiviral therapeutics. And we see here that binding of a known antiviral drug, vidarabine, into to the 3D structure of NSP16. And again, you can see here there are multiple hydrogen bonding interactions with important residues of this particular protein, NSP16. And vidarabine, obviously, you might be knowing that it is already a very, very important drug. And if you see here on the right side panel, we have shown the cavity view, where the molecules, how the molecules bind to the receptor inside the 3D cavity. So, yeah. Now coming on to the next one, which is a very important target. This is called the main protease, or 3CL-Pro, or we also see say 3C like protease. This is the protein which is involved in the cleavage of multiple long peptide chain to multiple fragments and ultimate production of 11 different functional protein of coronavirus. So if we can block this particular protein, what we can block is the formation of multiple viral proteins, and that's why it is very, very important protein. So in our study, we have identified elatrofloxacin, this is an antibiotic of floxacin family. Cangrelor, I already showed for a protein. Capreomycin, naltimedin, and indinavir. Indinavir is again an antiviral drug. So these are the drugs which are predicted to bind to this particular CL-Pro or the main protease. An important point about this particular protein I would like to say is it is cysteine protease. So cysteine protease means it is having a cysteine protein active site, and that cysteine can be targeted for the design of covalent inhibitors. Covalent means that the inhibitors that can bind to cysteine residue covalently, irreversibly, and then completely occupying the binding pocket of this particular protein. Right. So this can be an important target, and multiple people are working on this. So here we show the binding of different molecules into the main protease. Main protease is a very important protein, and we have got the co-crystal structure for this particular protein. And I can show you here the docking score of the co-crystallized molecule, which is something around 8.02, while the molecules which we have identified have a much better score than the co-crystallized molecule. So elatrofloxacin, and the docking score is -9.15. The capreomycin, the antibiotic, so it shows the docking score of -9.54. The cangrelor is actually a platelet inhibitor. It's a PY2 receptor inhibitor, and it shows a docking score of -9.05. And all these molecules show a very nice binding, and also they show the binding to some similar residue in the active site of main protease. So similar to main protease, there is another protease which is called the PLPRO, and this is again a cysteine protease, and you can see here the docking of multiple molecules. So first we started with the docking of co-crystallized PTP molecules to see what is the docking score of this particular co-crystallized molecule, and we have identified that the docking score is about -8.25, while we have identified those molecules, encyclovir, folic acid, methotrexate is a known anti-cancer drug, and it is also used for rheumatoid arthritis as anti-inflammatory drug. And here we have also identified that this particular molecule can bind to the binding site of this dopamine protease or PLPRO. And you can see here the docking score is -12, which is very, very high docking score. So the next one, and the in terms of drug design and discovery, I would say this is a star target, which is the RDRP or RNA-dependent RNA polymerase. And what happens, the primer is synthesized by NSP8, and ultimately they form a replication complex where there are three particular proteins, NSP12, which is RDRP, and then two more, NSP7, NSP8. Right? And as I said, this particular target, the RNA-dependent RNA polymerase, as the name suggests, polymerase. So this helps in the replication of the virus, and in simpler terms, we can say this is the photocopier machine. This creates the copies of the genome. So this is very, very important target. And in our analysis, we have identified cobicistat, capreomycin, then some antiviral drugs, elbasvir, indinavir, and remdesivir. Remdesivir is obviously it is a very well-known drug which targets the binding site of RDRP and stops the replication of the virus. And currently, this particular molecule, remdesivir, is under clinical trial. So here is the talking of our molecule onto the remdesivir. So the remdesivir here is shown on the, if you see on the left side of your screen, the remdesivir is shown in magenta sticks, while the molecule doxazosin, which we have identified, is shown in yellow sticks. And you can see here the molecules are quite well superimposed in the active site of RDRP. On the right hand side, you can see the 3D view of the cavity where the molecules are binding inside the receptor cavity. So you can see here how these molecules are stacking up inside the receptor cavity, and all these molecules are showing very nice docking score, that is at least -8.5. Then comes the other targets, the NSP15 or endoribonuclease. What it does is it cleaves the 5' 3' of UV-dilate residues in RNA by forming 2' 3' cyclic phosphodiester knot. Its activity can interfere with the host immune response and mask the exposure of viral double-stranded RNA to host the sRNA senses. So as I said earlier also, for NSP16, similar to NSP16, NSP15 also helps in masking the exposure of viral RNA to host immune system. So if we can block these proteins, especially NSP15 and NSP16, we can make virus more sensitive to our immune system, and that is why it is a very, very important target. And in our analysis, we have identified drugs such as octreotide, it is a peptide molecule, desmopressin, mesimorelin, and simprevir. They were found to target NSP15. So you can see here on the left hand side the docking of mesimorelin. Docking score is -8.5 with NSP15, and you can see here the drug is shown in green sticks while the receptor is shown in cyan color, and magenta dotted lines, they show the hydrogen bonds. So you can see here this particular molecule, mesimorelin, it is binding very well to the active site of NSP15, and on the right hand side you can see here multiple molecules which are inside the active site of this particular receptor NSP15. So overall, what we wanted to see is what kind of molecules are there, or how many molecules are there which can actually bind to multiple receptors. So if you see here, this is a heat map which we have generated. So on the y-axis, you can see the name of the proteins, nucleocapsid membrane protein, endonuclease, and up to PL-Pro, which is the papain-like proteins protein. And on the x-axis, you can see there are the name of the drugs, venetoclox, folic acid, nilotinib, and so on, up to cangrelor. And the scale, the docking score scale is showing here from 0 to -15. If you see here, the red one is actually better than -10, and the blue one is better than -8.5. So if you see here for the molecule cangrelor, so the cangrelor is binding to multiple proteins, especially RDRP, helicase, PLPRO, and hexanuclease, as shown by the dark red squares. Similarly, you can see here the methotrexate. Methotrexate is also binding to PLPRO and helicase protein, and there are multiple molecules which are binding to various proteins, as shown by the different colored squares. So the overall analysis shows that there are multiple molecules. We have identified multiple molecules which can bind to multiple viral proteins. And being a pharmacy student, you must be knowing that if you can target multiple targets in a disease pathway, that is much more effective strategy than targeting one single protein. And that was our driving strategy for this particular analysis, to identify molecules which can target multiple proteins in this particular disease pathway. And we have identified some molecules which can target individually multiple targets, or if you can combine those molecules, one or two molecules, then you can actually target more than 2-3 targets very successfully. So this is what we have done for the viral part, and these are the molecules which we have identified so far. And similarly, we used a database which is called the drug gene interaction database, because now we know from our computational analysis, as I shown in the first slide, that we have identified differentially expressed genes in COVID-19. And so we know what are the human targets or the host proteins which are getting affected by the COVID-19. So then this is a representation which shows there are multiple molecules which can bind to human targets as well as viral targets. For example, methotrexate, which is an anti-inflammatory drug, it is also very well known DHFR inhibitor anti-cancer drug. It binds to NSP proteins, and it also binds to human proteins. So here the viral proteins are shown in pink squares, the drugs are shown in blue hexagons, and the human proteins are shown in green colored spheres. So similarly, you can see here paclitaxel. So paclitaxel is binding to nucleic acid protein, and it can also bind to FOS and TUBB4B of human protein, which is a human protein and which has important role in the infection process. Right? Similarly, you have here lanreotide, which is a peptide, and it binds to multiple targets, NSP 15, 13, 10, 16, and it also binds to SSTR5, which is a GPCR. So this is a very important molecule. So the whole purpose of showing these particular molecules is that these molecules are actually multipotent molecules, right? They have multiple effect, and they can bind to various viral and human proteins, and therefore they can be very effective against this particular COVID-19 disease, right? Currently, what we are doing is we are trying to validate experimentally some of these molecules to see whether these molecules which we have identified, whether they are effective against viral proteins or not. So hopefully in the next few days we will be knowing that how many of those molecules which we have identified are actually effective against these viruses. So coming on to the conclusion that what we started the study with, we had the goal in the mind to identify the molecules that can dock with multiple viral proteins, and they can also interact with multiple host proteins. So basically, we started with the identification of the problem. So the identification of the problem told us that there are two parts of this particular disease. One is the viral part, the other one is the host part. So you wanted to target both of the part so that we can successfully treat this particular infection. And in this particular endeavor, we predicted multiple drug candidates, and we have taken a very high docking score of -8.5, which is roughly equivalent to 1 micromolar of binding affinity towards SARS-CoV-2 proteins. We hope that these drugs can be very effective because they are targeting multiple viral proteins rather than targeting single proteins. So because if you are targeting multiple viral proteins, the chances of developing resistance are very, very less rather than targeting single protein, because once you target single protein, the chances of getting resistance are very, very high. So our strategy from day one was to target multiple protein to alleviate this particular problem of drug resistant glutenase. So basically, in the last, I would like to say that our study predicts some promising agents that have potential to inhibit very crucial viral processes, upregulate antiviral host response, because they are also targeting multiple host proteins, so they can upregulate host response as well. And severe lung disease conditions can also be elevated. Especially, I would like to tell you about the molecule nebivolol, which is the spike protein inhibitor. It can also bind to ACE2 protein, and very recently it has been found that it actually stimulates the release of nitric oxide into the lungs, right? So what happens, this nitric oxide is actually dissolves the clots, and in COVID patients, especially the terminally ill patients, small blood clots which happens inside the lung are actually deadly, because there is fibrosis, there is already less surface for oxygen absorption, and if there are blood clots, then it can become a bigger problem. So this particular molecule can target viral proteins, can target human proteins, and also can improve the dissolution of blood clots. So therefore, we think this molecule can be very, very good candidate. Similarly, we have done studies for all those molecules. Apart from these docking studies, we have done extensive literature research, and based on that, we have identified those molecules. So these are the molecules which propose for repurposing, and we hope that some of those molecules will be ultimately found effective. So thank you very much. So in the end, I would just like to say that since currently we do not have any drugs or vaccines at hand, so I would urge everyone to follow government guidelines, use masks, use hygiene, and use social distancing, so that we can prevent this particular disease. Thank you very much. So I would be happy to take any questions.
Thank you, sir. Thank you so much. This is Ramlingeshwar Rao. Thank you, sir. So we request any of the participants to ask any questions to the sir. Just raise your hand, we will unmute you. Yes, I can see Kalam Mary Swarnalata. We are unmuting you, ma'am. Please ask your doubt. Kalam Mary Swarnalata. Kalam Mary Swarnalata. I request the technical team to unmute, ma'am. Yes, ma'am, you are unmuted. Hello. Please check. Deepankar Saha. Yes, sir. We have collected 4 more questions from the participants. Sir, shall I? Hello.
Yeah.
Sir, sir, you have shown minus 8.5 score in one slide. So which score, what is the score is considered best for knowing a drug to be very active? Is there any
yeah,
score like that?
Actually, there is no such gold standard, right? The best way is to dock a known drug and then compare the docking score with a known drug. That is one thing. But in our case, we do not have any known drug for COVID-19. So now what we relied upon is, we know that minus 8.5 in glide Schrodinger score, that is considered as a very good docking score. And in one of the paper very recently published, they say minus 8.5 roughly equals to 1 micromolar binding affinity. So that is starting point for a lead molecule. That could be a good lead molecule if we can identify with such affinity, and that's why we set the bar at minus 8.5.
Okay, sir. The second question from the participant. What is the software used by you during this study?
Okay, so we have used for molecular docking, we have used glide module of Schrödinger molecular modeling software, and for the analysis part we have used R and Perl script because the huge amount of data. We use these scripts, and for generation of those figures we have used PyMol and Schrödinger. And for gene expression analysis, we have used again R to identify differentially expressed genes.
Okay, sir. From a participant named Deshal, he is asking, besides Schrödinger, how is the Pyrex software for molecular docking and screening purpose used? Can be used.
So the Pyrex, or you are saying the Autodoc 4. So basically the Pyrex is the GUI for it. So this is a good software. The Autodoc 4 is good software, but you have to be aware what you are using. If you are using Autodoc 4, it is fine. But if you are using Vina, you have to be careful because Vina does not screen electrostatic interactions. It is just shape matching algorithm. It does very fast, right? So you have to see which algorithm you are using. Autodoc 4 is fine, very nice software. You can use it.
Okay, sir. Our next question from Poonam B. Is it possible to use the Gaussian software for the same study of molecular structure?
Why? Because Gaussian is a quantum chemistry program, right? So, and this is a very different purpose. This is a docking and other simulation. So in Gaussian, you cannot do all those kind of things.
Okay, sir.
The purpose of this is different.
Sir, what are the best softwares which are available free for the docking purpose? Freely downloadable.
So, very nice question. So there are multiple softwares which are available for docking. So there are, I will classify into broad categories. One is commercial, the other one is non-commercial. So the commercial ones, the good ones are, I would say, Glide is a very good one. Then we have Gold software from CCD, that is also a very good software. Then we have FlexX, then we have ICM. So these are all quite good software, very well-known software. In freely available softwares, I would say Autodoc is one of the best. So you can use Autodoc. Then you can use multiple scoring functions. So there are multiple scripts available online for free. So you can download and you can utilize those scoring functions to add into your Autodoc. And you can utilize multiple scoring functions to get much better hits.
This is a question from our college student, Sahitya Uppada. Sir, I'm a CSR NET JRF aspirant, currently studying the PharmD final year in our college. Is it better to attempt this after I pursue masters, or I can go directly for it? Which path is better? So as you have working with CSR also.
Okay, so the whole thing is want to do what? PhD.
Yeah, but currently she is in PharmD final year, sir. So that's why she was asking which path.
Okay, so most of the institutes like CSR institutes or DBT institutes or other institutes, so they admit students based on their fellowships for PhD degree. So if you have a JRF, whether it is a CSIR JRF, DBT JRF, or any other JRF, so that would be a plus point. You can get admitted to some of the national level institute based on your fellowship. So that would be a first starting point to get some kind of fellowship, any JRF.
Okay, Okay, sir. Our last question from the participants. Sharwari, he was asking, for students like us who wish to learn computational analysis, which institutes or courses would you suggest? Is there any institute or courses?
Institute, you can join any national level lab. You can almost all the labs provide training from 2 to 6 months or maybe more. So maybe you can join some of the national institute which is near to your place, and there you can get training for sufficient period of time, and then you can choose pursue your career based on your interest. Also, there are very nice online courses, but I would suggest to get into some lab, because it's not only knowledge, but it is the discussion with other peers and the feeling of that particular problem. So that only can be done when you are working physically in an institute with some people. So look into some lab which is near to your place and then try.
Okay, sir. Thank you, sir. Thank you for enlightening us. Over to convener.
Thank you.
Hello. 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 Thank you, Dr. Anshumanji. You have done good, fabulous informations to given our all participants. Totally 2,800, 400 participants are registered in this webinar. They today joined 400 members in at least Zoom and rest of there in YouTube. So all are given positive response. They taken good your speech, what your work done in your lab. Enormously, you are doing good job for the present pandemic situations. Really, I congratulate you are doing good for the public domain. So I'm in these junctions. I would like to thank you personally for your share your visions, what you are need for the research work, and also I'm thanks your acceptance our invitations to take in this platform. Your delivery your concept what is need to do for further research and under the Dreap. So in this, for token of our appreciations, I want to give your e-certificates by through Zoom today. Then later post by your mail. I request technical team to share the e-certificates on behalf of our management.
Awesome. This is looking awesome. Thank you very much.
Thank you, Anshuman. And now we want to have here in our college to give personal interactions by on another occasions. And because of again, I'm thanks to all. Because today is inaugural sessions, it may be late because some inaugural addresses are there. I really apologize for all the people who are participants. So tomorrow onwards will be in the right time. All the participants, please be give your any further communication through Telegram. We'll share the feedback link through chat box and YouTube description box and our college website. Please post your feedback. Tomorrow sessions will be at 4 p.m. Please kindly join at 3:45 itself, because tomorrow the session it is from US Dr. Sudhir Verma on the topics of bioinformatics approach for drug discovery and repositioning. He is a non-pharmacy actually. He is a biotechnologist and BTech. He is working in high cadre in high throughput principal analytic and consultant also. Please, all the participants request to join at 3:45 sharply. We have get good interaction with our international speakers. So, and today I would like to thank all our managements, Dr. Chairman Sir, Sri Jubrangraj Garu, and Dr. Masanagar, our secretary and correspondent, and all our organizing members, and Sri Sitaram Garu, OSD and Former Director, and Dr. Sunita, and all other HODs of the department, and fellow faculty members, fellow participants to join today's sessions. I really thank you, and thank you, Dr. Anshumanji, once again for this to taken this webinar part of the sessions. Thank you once again. I'm going to end of the sessions for today, and thanks once again. Thank you.
Thank you.